Hi everyone, I am Hiba Aribi, a master's student at University in Mana, Tunisia. I will present to you a bioinformatics study of transcriptomic and epigenomic changes in autoimmune demyelinating diseases. The main focus of genetic pathology studies are the differentially expressed genes as potential biomarkers or therapeutic target. However, the gene expression changes are controlled by epigenetic mechanisms such as the DNA methylation and the interfering microRNAs. In this study, we developed a pipeline to identify transcriptomic and epigenomic changes and correlate them. The workflow is as following. We first analyze the gene expression, the gene methylation, and the interfering microRNA expression data. Next, we develop an R package that will perform the correlation between the three data types. As output, it first identifies the differentially expressed genes, the differentially methylated genes, and the targeted gene of the differentially expressed interfering microRNAs. Second, it identifies the common genes between each two categories. Also, one of the package functions is to extract specific gene data. Multiple bioinformatics studies use publicly available datasets. Then they either combine or compare the results. For MS disease, we analyzed the blood cells data of European, Asian, and Australian patients that also represent three different prevalence areas of the disease. As a result, we found that the majority of the DEGs, about 30% of the DMGs, and 100% of the differentially expressed interfering microRNAs are population-specific. Next, we compared the data of MS and SLE disease of the same Chinese population to eliminate any environmental factor or genetic effect that could affect the data analysis. We found that the DNA methylation regulates 31% of the DEGs in MS and only 3% of them in SLE, while the interfering microRNAs regulate 30% of the DEGs in both diseases. Finally, we found that 14% of the DEGs in MS are at the same time differentially methylated and target of differentially expressed interfering microRNAs, while only 1% of the DEGs have a similar profile in SLE. We can summarize the results of the study in three major points. First, the developed R package that will be publicly available soon. Second, we must consider using datasets from the same population when studying the genetic pathologies. And finally, we believe that the study result could be useful to develop a new blood test diagnosis technique that will solve the misdiagnosis problem in similar diseases like MS and SLE. At the end, I should mention that the study was performed during the Omics Codathon, organized by the African Society for Bioinformatics and Computational Biology. So the authors, Hiba Aribi, Farah Hayedi, Karin Naitore, Olaitan Awe, and Sohaila Garbouch, thanks to the NIH, the UDSS, and the NCBI for their support during the study. Finally, I want to thank you for this opportunity and for your time. Thank you.
Okay, thank you, Eva. It was a beautiful presentation.
Thank you.
I want to make a question. Did you perform like quantify the efficiency or accuracy of this package you developed? Do you have a metric or something about it? How accurate is the prediction it makes?
The first analysis of the expression and the methylation uses standard techniques that are basically lemma and DSC. So then the identification of the differentially expressed and methylated gene is based on statistic, basically the p-value that must be less than 0.05. And then we obviously separate them between up and down regulated genes based on the log value FC.
Perfect. Did you have any, did you find any microRNAs or some sequences that you couldn't identify given annotation, an identification that you have to work on over them? or you could identify all of them?
There are some CPC sites that were not associated to the genes, and the majority of the microRNAs were associated. So it's only about the CPGs. We can improve that using the sequencing and the alignment. That would be a next step.
Oh, perfect. So your next step, it will be doing the alignment of these sequences. And have you thought about doing a kind of validation through other techniques, like, I don't know, real time, to know qPCR, to know if these microRNAs are really expressing, are really up or down in your system?
So as a result, we get, since we use the whole genome sequencing result, we get a big number of genes, and we also use it to diseases. So it's pretty hard to reproduce the same study in vivo. But we obviously get the list of the genes. The majority of them was already validated to be associated with the diseases in other studies that focus on only one gene or one microRNA. Minority of them were not associated, and maybe these genes will be studied in vitro.